Good afternoon, everyone, and welcome back to the Brainy Boomer Lecture Series. We're so very happy that you've all joined us today. In 2007, the McGill University Research Centre for Studies in Aging, or the MCSH Education Committee, started the Brainy Boomer Lecture Series in order to suggest practical steps to both improve and maintain brain health, as well as to promote healthy lifestyle choices amongst the most populous generation in history. The MCSH Education Committee, which was founded in 1996, has three main objectives: identifying education needs of healthcare providers, seniors, caregivers, and the public, and to develop responses to meet some of their needs; to enhance the positive image of the aging process by addressing stereotypes and myths about aging; and finally, the dissemination of research on aging. Dr. Pedro Rosa-Neto is a clinical neurologist with expertise in the quantification of dementia pathophysiology and preclinical diagnosis of Alzheimer's disease using biomarkers. He is affiliated with the Douglas Research Institute, the Centre Interuniversitaire de Santé et de Services Sociaux de l'Ouest de l'île de Montréal, and the Departments of Neurology and Neurosurgery, Psychiatry, and Pharmacology and Therapeutics at McGill University. He was appointed director of the McGill University Research Centre for Studies in Aging in 2017, which was soon followed by a professor position in neurology at McGill University in 2019. Before continuing, we would like to remind you to please keep your microphone on mute on Zoom, and that if you have any questions, you can either write them down in the chat box on Zoom, or you could ask them live at the end of the presentation. And now I'd like to invite Dr. Rosa-Neto to start his presentation.
So thank you so much. Can you hear me well?
Yes, yeah.
Okay, so thank you so much for the opportunity to speak to you today. So as part of this, of the Brainy Boomer Lecture Series, and I'm thrilled and I'm happy to be able to convey some news about what you're doing in Alzheimer's disease and what are the new concepts and new definitions of the disease. So I'm going to talk about Alzheimer's disease, the past and the future. So this is an ancient Roman coin with a deity and called Janus. So Janus was not adopted from the Greek mythology. It was something very Roman, and it was a double-headed deity that looks to the past and looks to the future. So it was something that could be found in the entrances of the houses, and the name of the month of January comes after this deity here. So this gives us the perspective from the continuity from the past and to the future, and this is something to do with what I will try to convey to you today. So we're talking about Alzheimer's disease and loss of memory, but before that, I think I have to give a perspective how the brain works. So what we call cognition is a summation of various different types of functions, like attention, like language, like the ability to write and to read, the ability to put things in order into a perspective in order to accomplish a task, remember things, and to orient in time and space. And everything together translates in our ability to cope with our activities of daily living, to get things going, interact with the others, and exchange feelings and thoughts with our peers. So the brain is very well organized in order to conduct this type of tasks. And in here, you can see that there is a particular architecture. So the cells in the brain, let me put the laser pointer, are organized in a very specific way in order to be able to perform all these tasks. So there are 100 billion neurons in the brain. Every neuron talks to the other at the frequency of 50 times per second. So all the cells here, they are connected by a very important pathway of projections of these neurons. And there is, and these neurons are integrated in the 1 trillion synapses. So they communicate with each other at this magnitude here, 1 trillion times. And in order to sustain this function, we need the brain, of course, takes a lot of energy, burns a lot of energy, and consequently produce a lot of waste. So in order to provide the energy, we have the arteries, right? And the arteries provide to the brain about 15% what the blood, or what the heart can pump. The brain takes 20% of the oxygen of the body and take about 25% of the glucose, the sugar that exists in our circulation. So we have also a very efficient system to clear the waste of metabolism, because there is a lot of energy that is produced. And of course, there is a lot of waste that is produced. And the cerebrospinal fluid and other systems of the brain allows us to remove this waste in a very efficient way. So now, from the normal cognition to dementia. Dementia is a very old concept. It was coined by the French alienists and doctors. And this is a name that stands out, Jean-Étienne Esquirol. So he was working in one of these hospitals for people with mental diseases. And since a long time, dementia has been recognized as a condition in which there is a decline of a previous level of functioning. So people were working, and then there is an important decline of the way they work. So it has to have the decline in two, for example, memory, or for example, the ability to speak and to remember, and to write and to read and to understand. And it has to interfere in the ability to function at work at the usual activities, and cannot be explained by any kind of other cause, like excessive intoxications, or perhaps overdose of medication and other risks. So the question of mental disease and the question of dementia was very important in the end of the 18th century. So this is a picture that depicts some German scientists that were very preoccupied and very devoted and motivated to find the source of the brain. And here we can see Alois Alzheimer himself. Here is Kraepelin, is the mastermind, the person who would find funding to do this research. Here is Gaup, and here is Nissel. So these doctors and scientists from the end of the 18th century, they had the dream to find the source of madness in the brain. And at that time, the technology that they had was the microscope. The microscope and the microscopic techniques, they have just been invented. And they discovered a lot of dyes that could reveal the secrets of the brain. And at that time, the vision that they had the brain was something like this. There was some fibers, there was some cells, and they believed that the madness of the mental disorder would be a consequence of a disruption of the normal anatomy of the brain. So they were expected to find something wrong in this structure that could explain psychiatric symptoms. So they cut the brain of patients with schizophrenia. They didn't find anything. They cut the brain of patients with depression. They didn't find anything. And many other psychiatric disorders and conditions, and didn't find anything. One day, this is Alois Alzheimer's with his co-workers. He found a case that he thought was very interesting. It was a female with 51 years old, and this was back in 1901. She was admitted by memory problems, language deficits. She was incapable to work anymore. She had important psychiatric symptoms. She became very paranoid. She was apathetic. She didn't want to do anything. She didn't have any motivation to do anything. And after the admission, her disease progressed, and she died a few years later, after 5 years of evolution, and she died because of a septicemia as a consequence of infections in skin. So when she died, Alzheimer was not even working in the same hospital. He came back actually from the city where he was working, and he took care of the evaluation of the brain of this lady. So he found in the brain, and these are the original pictures from Alzheimer's, that there was a deposition of something between the neurons. At that time, this was well known, and it was called a miliary foci. But he also described in the same lady this type of a formation here, which is something that accumulates inside the cell, where he named as a neurofibrillary tangles. And he also noticed that in the brain of these patients, there was a significant amount of atrophy. So Alzheimer, saved the day, because, the amount of money that was invested to find the source of madness in the brain somehow failed until the day Alzheimer's found this specific case. And he described a disease that he called early onset dementia, or pre-senile dementia. And Alzheimer's disease at that time was considered a very rare disease present in the young patients with these alterations in the brain. So then it came the war, the first one, the second one, and then, there came various changes in the social organization, improvement of our social conditions that increased dramatic the life expectancy. So this is the life expectancy, around the time of Alzheimer's disease. This is a chart from Statistics Canada, showing the life expectancy about 60 years old by 1920, and now the life expectancy that reached above 80 years old for females. There was a big change. So the elderly population, they expanded, and because of that, and because of the therapies for diabetes, therapies for high blood pressure, and high cholesterol, the population could age better and healthier for much more time, and therefore, diseases that were not very well known at this in the beginning of the century, last century, start to emerge by the 60s. So when Alzheimer's disease turned from a rare disease to an epidemic, so from 1910 to 1967, it was considered a very rare disease. But thanks to the work of some British neuropathologists that were interested in the aging brain, they realized that the findings that Alzheimer described in this young individual were very common in patients over 65 years old. And this started actually to raise concerns and questions about what is the meaning of these pathological findings in the brain. And it came actually in 1976, thanks to the work from Robert Katzman, that disease became the Alzheimer's disease became a much more important problem, and probably the most important cause of dementia present in patients after 1970s. So in 1984, it was coined the first operational definition of Alzheimer's disease. And this operational definition, links the concept of Alzheimer's disease to dementia. So this operational definition was somehow proposed because although most of the cases had the diagnosis of Alzheimer's disease at that time at postmortem, there are some cases that were not necessarily found the neuropathological changes of Alzheimer's disease, meaning accumulation of amyloid and tau. So this was a very important mark that somehow identify Alzheimer's disease as the most important cause of dementia in patients over 65 years old. So this definition remains nearly 25 years, and no progress in terms of therapy was reached. And it came due to research that by the time the patients develop the symptoms of dementia, there is a substantial amount of damage in the brain. So there was the need to change the definition of Alzheimer's disease in order to pick up phases of the disease, in order it would be possible to come up with somehow of intervention, or somehow preventing the disease, or somehow changing the course of the disease from normal cognition to dementia. So then, in 2011, the National Institute of Aging and Alzheimer Association created a new concept of Alzheimer's disease. And the concept of Alzheimer's disease came to the development of biomarkers, which are tests that are capable to detect the presence of amyloid and tau in the brain even before the beginning of the symptoms. So in 2011, Alzheimer's disease left the realm of dementia and became a disease characterized by a progressive accumulation of amyloid and tau. And this progressive accumulation of amyloid and tau, they were associated with 3 clinical stages. A clinical stage that is characterized by no symptoms and the presence of these 2 proteins in the brain. The second is a phase in which it is characterized by some symptoms, like memory problems, but they're not sufficient to meet criteria for dementia, and the dementia phase of the disease. And this was extremely important because this operational definition was much more lenient in terms of symptoms. So this is a summary of the 3 phases of Alzheimer's disease. So this is what we expected in the normal aging, because the normal aging always comes with some degree of cognitive decline. And here in this dotted line, it represents the decline characterized by the dementia of Alzheimer's disease. So the preclinical stage, where patients are nearly normal and they have the disease in the brain, was characterized initially by the publication for Ray Sperling. Ronald Peterson characterized the mild cognitive impairment, and Guy McCannon characterized the stage of the dementia. So this classification also incorporated patients with genetic forms of Alzheimer's disease, like patients carriers of autosomal dominant Alzheimer's disease. It also incorporates patients with Alzheimer's disease associated with the Down syndrome, and also recognize, very importantly, other forms of Alzheimer's disease. So this is a schematic representation that show pictures of the brain scans with the position of tau in various different regions. So this is to show that there is a type of Alzheimer's disease that has amyloid and tau in the brain, and is characteristic by agnostic symptoms. So this is the vast majority of patients that we know. But we always have to remember that there are other Alzheimer's diseases that are characterized by other symptoms. This is the visual spatial Alzheimer's disease, and you can see that there is an accumulation of the disease in the areas of the brain that we need to use to recognize things that we see, also to recognize, to write, and to read, and also to recognize other individuals. There is also a language variant of Alzheimer's disease where the most important symptom is the difficulty to find words. So these individuals here, they cannot speak properly because they lose the thread of their thoughts because they are always searching for a word to add in the sentence. They know exactly what they want to say, but the word doesn't reach to the mouth. When you measure their memory is not that bad. Their problem is much more located at the level of language. And there is another type of Alzheimer's disease in which memory is not that much affected. And the patients complain the inability to place things in order to accomplish a specific task. They have difficulties to cook, they have difficulties to set the table, they have difficulties to conduct some tasks at home. And this happens because the proteins that causes Alzheimer's disease are located in different brain regions. In patients over 65 years old, the amnestic type of Alzheimer's disease, the one that we know, is much more frequent. But in young patients with a younger than 65 years old, the prevalence of other forms of Alzheimer's disease is much larger as compared to patients over 65 years old. And with advent of novel biomarkers to visualize tau, the protein tau in the brain, and the ability to conduct, identify biomarkers at large scale using blood samples, it makes the diagnosis change again. And in 2018, Alzheimer's, the symptoms, they just left the classification, the definition of Alzheimer's disease. And since then, Alzheimer's disease is characterized by the presence of amyloid, the presence of tau, and the presence of neurodegeneration, what we call atrophy. Symptoms are part of, they work as a complement for this new classification. And this is a very important change in the way we see the disease, because Alzheimer's disease left the idea of dementia and became a disease very similar to hypertension, in the sense that hypertension is a state in which you have high blood pressure. And if you don't treat the high blood pressure, you will have in the future other problems like stroke or cardiac attacks. So when you think about the hypertension, what you want to do is the prevention of the consequences of the high blood pressure for a long period of time. In this specific case, what we're calling is Alzheimer's disease is a disease of aggregation of two proteins, the protein amyloid and the protein tau, which aggregate over time. And the idea is that if you stop this process of aggregation of these proteins, you can avoid the pathway from the normal cognitive state to the dementia state. So the way we talk about Alzheimer's disease today is when the patient has no amyloid in the brain, has no tau in the brain, and has no neurodegeneration in the brain. So this is A minus means no amyloid, T minus means no tau, N minus means no neurodegeneration. We call this a normal person. So if the patient starts with amyloid in the brain, we don't call it yet Alzheimer's disease. We call it Alzheimer's disease pathological change. And if the person has amyloid, has tau, and has with or without neurodegeneration, we call this problem as Alzheimer's disease, independent if the person has symptoms or not. So this is extremely important because this allows therapy of individuals with this condition in the brain and allows us to employ ways to remove these pathological changes in the brain and in the attempt to avoid and to prevent dementia. Because when the patients, they have amyloid and tau and no neurodegeneration, a bit of neurodegeneration, there is the hope that the brain itself will be capable to rewire and to survive or to the pathological process in the brain. So when you do a neuroimaging, so this is what I would like. These are the types of outcomes that you can have in patients and in many different age groups. So here is an image from a cognitively young individual with 24 years old, completely asymptomatic. So here are 3 cuts in the brain. So this on the top, we cut the brain like this, we call axial slices. Here, what you have is sagittal slices. You are cutting the brain on the anterior to posterior axis. And here we have the coronal slices where you cut the brain like this. So the way you read the images is like this. Hot colors means a lot, cold colors means a little. And you can see that in the brain of a normal individuals, there is basically no presence of Alzheimer's disease pathophysiology. Here is a scan of a patient, a normal person with 78 years old, a male. He got a Mini-Ment of 30, means the cognition is fine. And what you can see here is that this is the tau scan, and you can see that there is no tau in the brain. All the colors here, you can see they are in the very low range. Whereas in terms of amyloid here, you can see that there is a lot of amyloid in the brain. So this means that the person has already started the path to develop Alzheimer's disease. And this is a process that we expect to see in the brain of patients about 15 to 20 years before the beginning of the symptoms. So this is the first event that happens in the brain. And this person, of course, is completely asymptomatic. Here an example of somebody who meets criteria for dementia. It's a female, MCI, sorry, mild cognitive impairment. She has a cognitive alterations, probably involving memory and orientation. And here you can see that there is a lot of amyloid in the brain, just like here. And contrasting to this image here, we can see that there is some spots of very hot colors, meaning that there is tau in the brain. And the tau in the brain is affecting the brain regions that are subserving the memory function. So there is, when you see, if we have amyloid, there is not necessarily a correlation one-to-one between the presence of amyloid and the symptoms. But when tau arrives in the brain, it means that it starts to cause problems. And here it is an example where you have a bit of symptoms and a bit of tau. So this is a patient with 55 years old, it's a male, meets criteria for dementia, and we can see that there is amyloid in the brain, and you can see that there is a lot of tau in the brain. So the presence of protein tau, the location of protein tau, and the magnitude of protein tau is a hallmark of the symptoms. It's just like you could see whether somebody is symptomatic based on the presence of tau in the brain. And this is extremely important because if you can use these techniques to identify the presence of this pathology in the brain, we can devise medications capable to remove this pathology in the brain in order to keep the brain healthy. So another important development is that we realize that the dementia is not, that Alzheimer's disease is not the only causes of dementia. So there are other types of proteins that they aggregate in the brain, just like amyloid and tau, and they cause different types of symptoms. For example, Parkinson's disease, you have aggregate of another protein called alpha-synuclein that accumulates in the brain and also becomes toxic and kills the neurons. We have amyotrophic lateral sclerosis that is somehow is a protein that aggregates and accumulates in the brains, in the brain regions responsible to execute motor functions. And the brain individuals, they become atrophic and they die as a consequence of the disease. So recently, you know, another disease that has exactly the same symptoms as disease, we call it late, called the limbic predominant age-related TDP-43, has a very ugly name, but this is a disease that is characterized again by a protein that aggregates in the brain, accumulates, and kills the neurons. And we have a cortical basal syndrome, which is the disease that also affects the motor symptoms and the ability to execute motor functions. We have Huntington's disease, we have frontotemporal dementia, have Lewy body dementia, and we have also the primary age-related tauopathy, various diseases. But what's important for you to understand from this is that there are two major causes of dementia, and there is also vascular dementia. Is the dementia can be a consequence of a circulation, of a problem that accumulate to deliver to the brain nutrients and oxygen. And the second cause of dementia is the accumulation of toxic proteins. When the toxic proteins are amyloid and tau, we call it Alzheimer disease. When it's alpha-synuclein, we call it Parkinson's disease or Lewy body dementia. When the toxic protein is TDP-43, we call it late, or they call it frontotemporal dementia, or they call it amyotrophic lateral sclerosis. So there are two basic mechanisms that make somebody progress from normal cognition to dementia. First one are these proteins that aggregate in the brain, and the second one are problems related to circulation. How can you detect this? So how can we understand and how can you pick up the symptoms at the stage of the disease that you can really do something? So we develop, and the scientists develop something called biomarkers. And the biomarkers are chemicals that you can capture using imaging tests or tests on the blood or the tests with the cerebral spinal fluid that reveals the presence of these various pathological processes in the brain. So here you can see how this looks like in terms of imaging. This is a representation of the brain with a lot of hot colors, the position of amyloid in the brain. In this case, the protein tau in the brain. And when the patient have these two pathologies in the brain, you call Alzheimer disease. So this is a marker that you can see neurodegeneration. You can see the glucose metabolism, a test that we use to detect the presence of alterations in the neurons, shows some spots indicating the lack of function in the brain. So this is an indicative of a neurodegenerative process. A DET scan that allows us to detect Parkinson's disease and Lewy body dementia, and also an MRI that allows us to identify regions in the brain where you have atrophy. So, and these are various types of markers that you can identify in the brain. So today, with these tests, you can tell whether somebody is developing Alzheimer's disease. At some extent, with individuals are developing a Lewy body dementia. And these tests are currently available, especially in the research context. So because at this point, because you don't have yet a medication available today that we can offer to the patients, these biomarkers and these tests, although they are showing the presence of the disease, they are not necessarily, they don't have necessarily an application today. So the science is going behind actually the therapy, and we're going to discuss a little bit more in the next few slides. So this is an important development, which is a medication called Aducanumab. Aducanumab is a disease-modifying intervention. What does it mean? It means that this medication removes amyloid in the brain on the hopes to bring a cognitive health to the patient. So the idea is that by stopping the pathological processes in the brain, by stopping the accumulation of these toxic proteins in the brain, the idea is that the patients will improve from the cognition. So as you know, and as you heard, and as you read, there is a lot of controversy about that. So because we are in a moment of a transition from classic medications, symptomatic medications to disease-modifying interventions, there is a lot of questions whether giving a medication that removes amyloid in the brain will be somehow beneficial clinically for the patients. So this was an important publication showing a patient before the therapy with this Aducanumab drug and after therapy with this Aducanumab. And you can clearly see that the hot colors that indicate a lot of amyloid in the brain, they get blue. And this essentially tells us that amyloid was removed. And they also noticed that these are the patients before the medication, and these are the patients after the medication. That the individuals that they receive more medication are the ones that they get less amyloid in the brain after the therapy. And there were essentially 3 studies looking to the drug. This study here show a positive effect on cognition, but the 2 subsequent studies, one show an effect in the cognition and another do not show the effect of cognition. But the bottom line is that these medications, they do what they're supposed to do. So here is a plot demonstrating the amount of amyloid in the brain. This dotted line shows the difference between abnormal amyloid and normal amyloid. And you can see that in patients that they received 10 milligrams of Aducanumab on the course of 78 weeks, this medication was capable to remove progressively amyloid in the brain and brings back what was expected to see in normal people. So does it help patients clinically? We don't know yet. So we will know pretty soon, but the truth is not yet accepted for known. So the summary here is that to show what we see Alzheimer's disease in the past. In the past, Alzheimer's disease was a kind of dementia. And with this framework, it was impossible to find a medication or an intervention to rectify the cognitive abilities of people, simply because the brain is already too destructive, too damaged when the patient gets to the point to develop dementia. In 2011, the Alzheimer's disease became a disease characterized by accumulation of amyloid and tau in the brain with three clinical stages. The preclinical stage, where there is the proteins, but not the symptoms. Mild cognitive impairment, there are some symptoms, there are the proteins, but the symptoms are not enough to declare somebody with dementia. And the third phase of the disease, which is characterized by dementia. And in 2018, we completely dissociated the symptoms, the disease, Alzheimer's disease, the symptoms of the disease, and Alzheimer's disease became something like hypertension. It's a disease in where you have to remove these aggregates of bad proteins in the brain in order to keep the brain healthy. So we here at the center are working very hard to make the diagnosis available for people as soon as possible. And we are working very hard. We were the first center in Canada to implement tau imaging. So we're able to see this tau in the brain, and we use this tau in the brain in order to, as a metric, to test biomarkers, to test blood tests to identify the disease without the need of the scanner. And we are doing this because it's a very expensive procedure to scan somebody. So it would be much better, much faster to obtain the diagnosis using a simple blood test. So we're very happy actually to collaborate with our group in Sweden. So the papers that we produce here in Canada in conjunction of them, yield a lot of interesting, important publications. He's Nick Ashton. He received a prize from the Queen of Sweden with one of the publications that we did together. So this is Thomas Cagliari. Is also our awardee that received a prize as a consequence of the publications that we did together with them. So and this morning I asked from one of our associates, Andrea Benedette, if she had a picture like this. She couldn't send me the picture because she was working with our samples this morning, and she was developing a new technique for early detection of Alzheimer disease. But she showed a picture of herself this morning, work with the sample. This picture was taken this morning in Gothenburg, Sweden. So and this research was obtained by your help. So this is our cohort. This is the triad cohort. If you go to our webpage, you can see the testimonials of our participants telling what happened and how do they feel and how do they perceive our work. So it's a very touching testimonial. I advise you to look at it. And here are the numbers of the cohort. So all these people here, way more than 1,600 individuals, they have all these metrics in the brain. And because of these metrics in the brain, you can develop Alzheimer new biomarkers to identify the presence of the disease in very early stages. And again, to identify the disease in the silent stage of the disease is a precondition for therapies able to avoid the brain destruction and lead a person to dementia. So our outreach program is also something important, because what I'm telling you today, and to diffuse this information and to bring these things to the debate and to answer these questions and to involve you in these studies and in the progress is something that's extremely important, is under the heart of our research program and disseminations program, in the heart of the MCSA. And I'm very happy, and I'd like to thank all of you to be part of this process. So we had 2,327 participants since January during the 35 Brainy Boomer lectures and 35 exercise for seniors, which are the programs that we're developing here in order to engage, in order to exchange the ideas, and in order to get some inspiration from you to move forward in our research. So I think one of the highlights is that we attract an astronaut. So Jay Buckley, he came here to give a talk, and he is devising again, new techniques, and he doesn't want to do a blood biomarker like us. He's much less invasive. He wants to develop, he wants to detect the presence of Alzheimer disease by simply giving a test where people have to listen some auditory stimulation and able to discriminate what's been said on the using some phone headsets. And this is incredible because the idea is to bring actually less invasive technologies, much more sensitive technologies, to be able to come up as early as possible with interventions and be able to make that these interventions, they are causing benefits to the brain and not harming the brain. So early this year, Dr. Gauthier, who is now an emeritus professor, we miss the presence of Dr. Gauthier here. He came up with another level of knowledge translation, which is to bring these things that we are discussing today with you to the world. So Dr. Gauthier is the chief editor of this Alzheimer disease report, where we, as you can see, the cover is very telling. what you're seeing here is the invisible part, the invisible phase of the disease. And this is essentially conveying that if you want to eliminate dementia and if you want Alzheimer's disease, you have to treat early, treat asymptomatic patients, and treat the disease based on the presence of the pathology in the brain. This is a completely different change in the way people see Alzheimer's disease and the way we think about Alzheimer's disease, because we are disconnecting more and more Alzheimer's disease from dementia. And dementia becomes a very late stage of the disease that starts 20 years before. So this is the third level of action that we're taking is in terms of developing new technologies. So next week we are bringing here the creme de la creme of people developing biomarkers in order to discuss the use of biomarkers in the brain and applications in many different contexts, not only in Alzheimer's disease, but in other neurodegenerative psychiatric conditions. So the MCSA is leading local and international initiatives in order to combat Alzheimer's disease. And also for our local populations here and for our associates, we are bringing actually the knowledge translation at the maximum level. This is a series that the center developed for students and healthcare professionals. And we are talking about a series of different events. We are talking about, this is an event that aims to understand the details of protein aggregations like amyloid and like tau as a driver of a cognitive decline. And tomorrow we will bring here the Nobel Prize, the guy who actually came up with the idea of protein aggregation and misfolding as a source of the disease in the brain. So I think the center is also heavily contributed and heavily committed at locally and internationally in order to develop ways and in order to develop techniques to early detections and to treat patients with these conditions. This is another very nice example of the success of our student. This is Joseph Theriot, who is just received the 30 under 30 from Forbes, and he received this prize given to his research on detecting the disease in the brain of patients at risk to develop Alzheimer's disease. So he's a student here from our center. And the action doesn't stop there. We'd like also to bring to the patients not only information about the disease, but also information about changes in health styles that can improve patients and mitigate the process to develop disease pathophysiology. And this is an example in the form of the cookbook. So I'd like to tell that the center does this for you. So we'd like to extend our gratitude and appreciation and for you, for our patients, for caregivers, for our research participants, for our colleagues and partners. And I'd like to tell you that you are always in our past, you're always in our present, and you're always in our future. And we would like to thank you very much for the